about her recent and further back work in the genomics of adaptive evolution. And this talk is part of her going out for promotion and tenure. And so Molly started out at UCLA, where she got her bachelor's degree. Then she went on to UC Irvine, where she worked with Michael Rose, who was kind of one of the founders of the study of the evolution of aging. And there she used these long-term selection lines in Drosophila to test a very fundamental question about how adaptive evolution proceeds at the genomic level in outcrossing eukaryotes. And up until then, it was thought that, and I hope I'm not breaking into what you're going to talk about. It was thought that major adaptation involved the selective sweeps of brand new mutations. And that was mainly because everybody worked on bacteria. And this was the first time somebody had used an outcrossing eukaryote to test the same, whether the same thing happened. And she found something completely different. That in fact, adaptation involved multiple loci spread throughout the genome and standing genetic variation rather than brand new big mutations. And this was big news. Her first this chapter out of her dissertation was published in Nature. And she then went on to do a postdoc with Anthony Long, also at Irvine, where she continued to ask similar questions, but she developed this novel system using yeast as a model outcrossing eukaryote, which she'll tell you more about. So here she teaches genetics, evolution, evolutionary genetics. She's mentored a number of graduate students, including 3 postdocs, including our own Mark Phillips, who we were smart enough to hire as soon as he became available. And so today she's going to tell us about the expanding work with her system.
Thank you. What a nice introduction. That was awesome. And it's so awesome to just be here and have an opportunity to tell you about the work that I've been doing since I started my lab here at OSU. And because I might go long on time, I thought that I should first just say how profoundly grateful I am and how fortunate I feel to have gotten to start my lab here at OSU. It's been such a wonderful intellectual environment to do the work that I've been doing. And I've had such wonderful trainees who have really put a lot of blood, sweat, and tears and joined me along this journey. And so thank you to everyone in the lab, past, present, and future. And like Mike said, maybe particularly Mark Phillips, who is now, of course, assistant professor here, but did a lot of work in the lab as a former postdoc, and that work is going to be featured throughout the talk today. Okay. So I tend to start talks with this statement, which I think will be non-controversial to this audience, but basically, I think that evolution is really hard to study. It's hard to study empirically because if you think about it, all the species that we can observe on the planet have evolved exactly once. And so if we want to try to figure out what happened in the past to shape populations as we see them today, we have to rely on the fossil record, which is notoriously incomplete, or maybe ancient DNA, which is hard to come by. But really, we're restricted to making comparisons across sort of extant lineages. And that's a very indirect way to study the process of evolution. And so famous evolutionary biologist Stephen Jay Gould posed this famous thought experiment in his book, Wonderful Life. What would happen if we rewound the tape of evolution? This is like imagining an old VHS tape, and replayed it. What would happen? Would we get sort of humans as we know them today? He thought no. He thought that the role of randomness and stochasticity would be so great that there's no way we would get the same species to evolve twice. But the point really is we can't know for sure. We can never know. That is just a thought experiment. However, there are some techniques we can use to study evolution in the present tense in a predictive way. And one of those methods is experimental evolution, which is something that my lab does. It's a theme that unites all the projects that we've done in my lab. And so when I say experimental evolution, what might come to mind first for you is selective breeding, right? And we know that selective breeding works really well. Offspring look like their parents, and this fact has been exploited so that every time you go to the grocery store and look at the diversity of cruciferous vegetables, or the size of chicken breasts, or the diversity of dog breeds, that you might have in your home. We just know that selective breeding works. But importantly, when I say experimental evolution, I'm talking about something that's subtly but importantly different from just selective breeding. What I mean by experimental evolution is sort of the specific context in which you bring a population of model organisms into a controlled laboratory environment. You subject it to some novel environment, maybe a stressful environment, and you document what happens as a result of evolution. And you want to do this by measuring phenotypes and genotypes in evolved populations and doing that in a parallel manner across multiple replicate populations that are independent and never interbreed, but they're all experiencing that same environment. And in that way, we can distinguish between processes that we can ascribe to adaptation versus those that might have come about by random stochastic processes like genetic drift. And so I like to try to argue that experimental evolution, yes, it's a method. It is a tool that we can use in our labs, but it's also a subdiscipline. There's been this community of experimental, or those of us who call ourselves experimental evolutionists, this is what we do. We try to model the process of evolution empirically, to study evolution in real time, and to make connections between genotype, phenotype, and evolutionary process. And in that way, we're able to address questions about the past, present, and future of life. So I'm going to break down this talk into 3 parts. First, I'm going to spend a little time explaining to you our study system, because like Mike said, that's sort of a big part of what we do is our study system. Then I'll try to highlight a few projects that we've done along the way over the years, and then end with some future directions. So Mike actually gave a really awesome introduction that sets up perfectly what I wanted to explain, which is that most evolution experiments to date have used microbes, and especially E. coli. You can imagine this is really practical. E. coli have short generations. I mean, the longest experiments that have been done with them have spanned, decades, but tens of thousands of generations, so a lot of evolutionary time. They have small genomes, so they're easy to genome sequence, and they're cryopreservable, which is a really important point, because it means if you have an organism that's cryopreservable, you can build a living fossil record in the freezer. So you're not just limited to studying extant populations. You can go back in the freezer and literally revive any time point you might be interested in and use it in an experiment. So you can actually rewind the tape of evolution and replay it with an organism that you can cryopreserve. But at the same time, E. coli, sorry, but they're kind of boring. They don't have that many phenotypes you can measure. You could argue that the complexity of those phenotypes is not great. Probably the most common metazoan model that's used for experimental evolution is Drosophila melanogaster. And it's a wonderful model system for this type of research, except for that it has a 2-week generation time. So even under the best of circumstances, you can't really get that many generations of evolution under your belt with this system without investing serious amounts of real time. So the longest experiments that we have with Drosophila, I was lucky enough to work with them myself as a grad student, but in 30 years, we get something like a few generations of evolution, not thousands of generations. So there are some limitations just associated with the life history of the organism, but at the same time, we really want, ideally, I guess, as someone who is starting a lab, I really wanted to use a model that was eukaryotic, had some complex phenotypes that we could investigate, and really importantly, had sexual recombination. So it turns out that the more and more genome sequence data we get from these evolution experiments, the more it is becoming apparent that asexual microbes and sexually reproducing eukaryotes evolve via fundamentally different processes. So as Mike sort of alluded to, that was work that I did as a graduate student, and even sort of wrote a perspective piece synthesizing the current evidence about that point several years ago. And so I knew that as an independent investigator, I really wanted to pick, I had to choose wisely, right? I had to pick an organism that somehow married all of these attributes. And so that's where I arrived at the yeast, Saccharomyces cerevisiae, which is the same yeast that makes our beer and our wine and our bread, so the baker's yeast, extremely versatile organism, but also a very popular model organism for genetics. It happens to satisfy all of these requirements. It is a microbe, a single-celled microbe, but it is a eukaryote, right? And while in nature it's thought to not reproduce sexually very often, it is facultatively sexual. So it can be made in a lab environment to reproduce sexually under the right conditions. And finally, it's cryopreservable. So like the bacteria, we can create these living fossil records of experiments with yeast. And so I really have sort of invested a lot of my career developing this model system of sexually reproducing yeast to do this experimental evolution research. So it turns out that yeast have a huge amount of genetic diversity across the species. So even though it's just one species that does all these things for us, you can imagine that the various sort of venues of domestication that exist, like wineries, bakeries, breweries, etc., have provided for lots of domestication environments that have led to a huge amount of genetic diversity. And so yeast lineages have genetic diversity that in so much genetic diversity that about 1% of the genome, those sites are variable. So that's even more genetic diversity than we observe across human populations. And we have lots and lots of lineages to choose from to do this kind of work. And so in the lab, what we'll often do is we'll take strains collected from around the world or from different environments that are genetically distinct, and we will mate them, impose a series of sort of culture conditions to make them reproduce sexually and shuffle up their genomes so that what we produce is a population of cells where each cell is an individual, and those individuals each harbor unique genotypes. And there are hundreds and thousands of those unique genotypes in the population. And so in this way, we can create populations that harbor a great deal of genetic diversity and use those in evolution experiments to test questions about how that diversity evolves to any number of circumstances. So I'm guessing that, not in my lab, but those of you in the audience, there might be many of you who could use a refresher in yeast life history. So yeast can exist, it's very, it has a really complex life history. It can exist as a haploid or a diploid, and it can reproduce asexually or sexually. And so this figure comes from one of the methods papers we've published over the last couple of years that kind of illustrates a typical workflow in one of our evolution experiments. And so we can start with step one. We'll start with we'll start with haploid cells that might have different genotypes. We can put them together so that in a way to promote their mating, they attract each other with pheromones. And then assuming the right genetic markers have been engineered into them, we can recover the diploids using selected media. And those diploids can then be subjected in step three to any number of evolution experiments that we can imagine. For maybe that means adding something to the media or raising the temperature, something like that. And then in step four, we would then induce sporulation. So essentially by starving yeast, they undergo meiosis and they produce 4 spores, which are gametes. And those gametes will then, we sort of impose a couple of extra steps to make sure they get properly mixed up so that when they mature into haploid cells, they can mate randomly. And so this workflow is sort of contrived. I guess I'll just say I'm proud to say that I think that this workflow is something special that our lab does that not very, very many other labs do. And that's sort of hard to do in the yeast world. I mean, there's a ton of labs that study yeast genetics, but a small fraction of them study evolution, and then a smaller fraction of them are interested in outcrossing and sexual reproduction in yeast. And so I think that's why we've had an opportunity to publish these methods as standalone papers. And we've been able to advertise the products of these methods to the community. So when we do this type of workflow, we've been able to produce a wide range of populations that are highly recombinant and have different genetic properties that come in handy for these evolution experiments. So for example, the populations we use the most have either 4, 8, or 12 distinct founding lineages. And they're kind of present in relatively equal proportions across the genome. And these are all useful ancestor populations to begin selection experiments or evolution experiments with. And these are resources that we share with the community, and collaborators and other labs have asked us to use these. So because a lot of the experiments that I'm going to tell you about today share some common features methodologically, I thought it would be worth sort of explaining the bread and butter of our genomic analysis quickly. So the most common technique, not the only technique, but the most common technique we use in the lab is called pooled population sequencing, which means that we will extract DNA from samples of populations in order to submit those samples for whole genome sequencing, and the results of that sequencing gives us allele frequencies. So you can imagine this is pretty easy in yeast. You just pipette yeast cells, extract DNA from them, submit them to the sequencer. And if you have two populations that you want to compare, maybe those two populations are the ancestor and an evolved population, or an evolved population and a control population, the data that we get back are allele frequencies, where the depth of sequencing coverage is our denominator, and allele counts tell us what the frequency is of a particular allele in the population. And so we might see, for example, that in population 1, we get an allele frequency of this G allele, the single nucleotide polymorphism, that's different than what we would measure in population 2. And there are over 100,000 variants in the yeast genome that we can do this kind of work with. So for any one of our evolution experiments, we get large, really rich data sets, genomic data sets, that are amenable to lots of different kinds of analysis strategies. We can ask, how did populations change over time? How are they differentiated relative to controls? Things like that. And we can also ask, if we have repeated sampling of the same populations, questions about sort of the nature of evolutionary change. What are the shapes of allele frequency trajectories as they evolve to novel environments? So as sort of to explain the system, I'm going to go through in some detail one experiment that I actually started as a postdoc, and then when Mark Phillips came to the lab as a postdoc, he continued to work on it in my independent lab. And I think it illustrates a lot of the cool things about the system, so I'm going to invest a little time in explaining how it works. So this first experiment that I did made use of the 4 founder population. So 4 genotypes were all mixed up. We started with this recombinant population, split it up into 12 replicates, and those 12 replicate populations only experienced the steps that I explained in that rainbow chart. They didn't get any sort of anything else, because as you might imagine, even though yeast can outcross, they don't really like to. It's a little bit stressful for them in a variety of ways. So this was just an initial experiment to sort of demonstrate that the whole idea was going to work. And so these 12 replicate populations went through this outcrossing procedure every week for 18 weeks. We estimate that's about 540 asexual generations. And then what I did as a postdoc is I sequenced a few time points to try to determine what had changed over that experiment. And then when Mark came along, he sequenced all the time points so that we could learn even more about what changed. And so what we found, so I'm going to show figures like this a couple of times throughout the talk. And if you haven't seen one of these before, we call it a Manhattan plot because it kind of looks like skyscrapers across the plot. And so on the x-axis, we have genomic location. So in the yeast genome, there are 16 chromosomes. So each of these white and gray squares or rectangles represents a different chromosome. And then on the y-axis, we have a significance score such that a higher value means more significant or more differentiation over time. And so what we see are these sort of peaks of localized change, where every point on this figure represents an allele that changed over time. And the higher it is on the y-axis, the more it changed consistently across those 12 independent populations. And so we see about 5 regions that are especially interesting as a result of this analysis. And these are all regions where we might want to look at those locations in the genome and ask, what are the genes there? Might they have contributed to the phenotypes that we observed in these evolved populations? So we call those candidate genes. And if you zoom in on any one of these peaks, like peak C, the most significant one, we can really do kind of a deep dive and ask not only what genes are there, but what is the nature of the variation in that location. So like the most significant alleles in this region don't occur in a gene, but they do occur in a region known to be sensitive to an enzyme called DNase-1, which means that they may have a role in gene regulation because DNase-1 cleaves regions of the genome to make spots accessible to transcription factors. And there were some alleles that were occurred in a gene. In this case, this gene is called MLP1, which encodes a myosin-like protein associated with the nuclear envelope, which I have no idea why that would be important for outcrossing, but it's a candidate that we could look into further. And so this is, again, just sort of this proof of concept that this is going to work in terms of localizing areas of the genome that we can associate with adaptive change. And one more thing we can do is not only think about individual alleles, but groups of linked alleles, because alleles don't evolve in a vacuum, they evolve in linked groups. And so these colored dots on the bottom of this figure illustrate the results of our attempts to resolve that a little bit. So when we ask, for example, which of the 4 founding genotypes is the most represented in this significant region, we see that the answer is the red one. And the red one, in this case, belongs to a West African palm wine strain. So there was something about the genotypes of this West African palm wine strain that conferred a fitness advantage at this particular region. And so, I think that this slide illustrates how well we can connect genotypes to experimental evolution regimes or evolved phenotypes. And in this next slide, I'll show you what we learned that's a little more general about evolution, which is that when we sequence all this, when we sequenced all the time points of this experiment, and this was Mark's work that was published a couple years ago, and we ask, in this case, AI tools like self-organizing maps to condense those time points into sort of the most common shapes of evolutionary change. What we see, if we just focus over here, is that there are 3 common allele frequency trajectories. Among those alleles that change significantly over time, there were 3 shapes. And those shapes are were only roughly sketched out when we sequenced a few time points, but become quite well defined and much higher resolution when we sequence more time points. So you can see shape 1 is this initial stasis followed by increase. Shape 2 is a roughly linear increase. And shape 3 is an initial increase followed by a plateau. And we became really interested in this last shape, this increase followed by a plateau. And, these self-organizing map figures aren't scaled, but when we plot the raw allele frequencies, so these are all of the allele frequencies belonging to the replicate populations of the orange group, you can see that there is that increase in frequency followed by a plateau about halfway through the experiment. And that suggests that alleles that are under selection are not becoming fixed in the population, despite hundreds and hundreds of generations of strong selection. Instead, they seem to be maintained at something like 80%. And so, importantly, that's not really predicted by most standard population genetic models. It implies that there are other forces at play maintaining variation in the genome. Those might be things like balancing selection or heterozygote advantage or frequency dependent selection. We don't know, but whatever they are, I think that we are finding in our experiments that this pattern a lot. We're finding the long-term maintenance of genetic variation despite doing these long experiments with strong selection. And so that's, I think, hopefully I've been able to convince you with this first initial proof of concept experiment that these types of experiments with yeast not only teach us about the genetics of specific traits or selection regimes, but also provide important takeaways about how evolution works generally. So now I can transition to talk a little bit about some of the specific traits we've been able to study, and I'll try to highlight the students and other trainees who worked on these. And what do I mean by complex traits, by the way? By that, I mean traits that are that should have a polygenic basis, so lots of genes contribute to them, and also genes which might have a significant influence by the environment. And so, in other words, all the traits we might be interested in studying as humans. And so my approach to mentoring trainees has generally been to try to work with them early and find a trait that interests them for some reason, maybe related to their background or just their own research interests. And in that way, sort of work together as equal stakeholders in the project. So I tell them about my experimental system, which we've invested a ton of time into, and it works pretty well. And then they help come up with an idea for a trait to explore. And that's been working pretty well. So my first graduate student, Kislana Wing, was here at OSU a few years ago. And the trait that she identified as one she wanted to work on was freeze-thaw tolerance. So as I already explained, you can cryopreserve yeast, but we sort of asked the question, well, is that stressful, right? Yes, you can put them in the -80 with a cryoprotectant and revive them and they survive, but could it be that that's a stressful process? We would imagine it probably is, right? And so what Kislana did was she took our four founder population, broke it up into 12 replicates that all experienced the same treatment. And that treatment was multiple rounds of deep freezing and thawing after being archived with a cryoprotectant, which for yeast, we use glycerol. And so after these, so basically what this workflow looked like was she would take a tube of yeast, she would inoculate it with one of two concentrations of glycerol, she would assay cell viability before freezing, then she would freeze it, and then immediately after pulling it out of the freezer, she would plate it again to assay viability after coming out of the freezer. And then she would save some cells for DNA extraction and sequencing. And lo and behold, what she found was that cell viability was indeed threatened by cryopreservation. So we started out, and these were the same, we found the same results for two different concentrations of glycerol, but I'm showing you the results in 15% glycerol. So they started out at something like 75%. Only 75% of the cells survived freezing and thawing, but that fraction went up over time with experimental evolution. And when we looked at the whole genome sequences of these populations and looked at the variation, we saw this really nice Manhattan plot with only one skyscraper on it, right here on chromosome 15, which implies that this is a region of the genome harboring variants that perhaps confer a benefit for freeze-thaw tolerance. And when we zoom in on that peak, on that one region, first of all, it's a bit bigger than it looks. If you just look at the whole genome, it actually is about, it spans about 25,000 base pairs, and there are 16 genes in there. So we don't know which one or ones of those genes might be the ones that confer a benefit in this case. But we can ask questions like, well, what about the most significant alleles, the ones that change the most? Are they in a gene? And in fact, they are. They're in a gene called ALR1. That is a gene that encodes a magnesium ion transporter on the cell membrane. And so it sort of shuttles metal ions across the cell membrane. And I don't know why exactly that would help with cryopreservation, but it's conceivable that it could be involved in osmotic balance in some way, because cryopreservation involves osmotic stress. And also we found that there is one gene for which there's no known function in yeast. That's why it has a funny name, but there's a non-synonymous allele among our most significant. And so that means it encodes an amino acid change. So that would be an especially good candidate for one that might have a functional consequence. So perhaps this gives us an opportunity to further annotate the yeast genome when we get results like this. And then finally, it turns out that all of the alleles that were significant belong to one haplotype. In this case, the strain name is YPS 128. It's the only one of the 4 that came from nature rather than sort of a food industrial commercial setting. So it was isolated from the oak of a bark tree on the East Coast. So we could speculate that maybe this strain has experienced overwintering stress that could help it with freezing and thawing. Although at the same time, I think that deep freezing at -80 is pretty different from overwintering, but it's just an idea. And then finally, just to take some larger evolutionary lesson away from this, when we zoom in on the most significant allele, the variant that changed the most over time in all the replicates, we again see that it did not become fixed. It increased in frequency to a degree, and maybe the experiment wasn't long enough, or maybe once again, we've just seen further evidence that these alleles are maintained in populations despite strong selection. Okay, so the next trait that I want to talk about is intensity of ethanol stress. And now I can properly introduce Mark as a former postdoc in the lab who got an NSF postdoc fellowship to do this work that I'll tell you a little bit about. So we were interested in, so ethanol stress is a pretty well-studied stress in yeast because of yeast's role in wine and beer making, right? And production of alcoholic beverages. But we chose it because it's known to have a really polygenic basis. It is a complex trait. And we weren't so much interested in the ethanol tolerance of it all, or the ethanol stress in particular. We were interested in what happens if you vary that stress. If you have strong stress and a weaker stress, do you get the same areas of the genome responding to those 2 stresses? And so to do this experiment, Mark established 20 replicates that got high stress, media supplemented with 10% ethanol. So they are not very happy growing here in the red. 10 replicates, they get moderate stress. So that's 6% ethanol. They do a little better, but still not as happy as the control populations that got no ethanol stress. And so he started with this 12 founder population and did this experiment for 15 weeks where we had the outcrossing every week. And then we looked at sort of genome-wide variation in the beginning, the middle, and the end of the experiment. And what he found, well, I guess first, before I tell you what he found, just to explain our approach here, again, we were interested in figuring out, were the high stress treatments versus medium stress treatments promoting different genotypes? And in order to do that, we kind of need to think about identifying categories of significantly differentiated alleles. Because, for example, what if we get alleles that are significantly differentiated in all the treatments, high stress, medium stress, and no stress? Well, that would just imply a general response to lab conditions or lab domestication. And we wouldn't really, those sites wouldn't help us answer the question of interest. We also might get alleles that are significantly differentiated in high stress and medium stress and not the controls. And those we would call general ethanol stress SNPs or alleles. And then the ones we were really interested in are alleles that were significant in only one of the 2 stress treatments, either high stress or moderate stress. And in other words, if we observed if these exist, it implies that there are different trait architectures for these 2 traits, even though it's the same chemical, ethanol, that's stressing them. It's the intensity of the stress that elicits a response. And so basically we found, this is a complicated figure, but the take-home message is most of the change that we observed is that, I'm going to keep with the color coding here, is the blue type. So general laboratory selection. So most of the change happened in all of the treatments, regardless of whether they got ethanol or not. But we do see instances of treatment-specific alleles. So ones that are either red in this plot or orange in this plot indicate that they were special, right? They only occurred in that treatment. And there are some, it's hard to see with this figure, but there are some especially interesting regions, like maybe right here on chromosome 7, where we see alleles that are significant in the medium category, but they don't show up at all in the high category. And that implies that for some reason, there are alleles that are beneficial in medium stress that are not beneficial in high stress. And that was super interesting to us. And another way to think about that is if we, so if we look at all of the SNPs, all of the alleles that were significant in ethanol, regardless of intensity, and think about their change over time, we got the least amount of change in the controls, a moderate amount of change in the medium stress, and a higher amount of change in the high stress. That's exactly what you would predict under the expectation of additive genetic variance. In other words, if there's just a set of ethanol stress genes and they respond more strongly in the high stress and less strongly in the medium stress. But if you focus on the alleles that responded to the moderate stress, we see something interesting, which is that they changed a lot under medium stress and then went in a different direction in high stress. So this again is just kind of hammering the point home that we think we see some evidence where there are alleles that are beneficial in a moderate amount of stress and they lose that benefit in high stress. And so that sort of complicates our understanding of the genetic basis of this trait, and it implicates either epistasis, which means genetic interactions, and or pleiotropy, which means one allele having multiple effects based on the environment. And it also could explain why one thing we've been noticing in this experimental evolution community is that it's difficult to replicate results across labs. And this might be one of the reasons why, is because if you don't replicate exactly the same protocol as your neighbor, you might get different results. And so we think that this not only is interesting in its own right, but has relevance to thinking about experimental evolution as a predictive mechanism. So how are we going to predict how populations are going to evolve in the future, say, in the context of climate change, if we can't even repeat results across labs? Okay, and then sort of the last trait that we've been doing some work on that I'll talk to you about today, in this section anyway, is reproductive longevity. So we've been using yeast to study aging and late life fertility, which has been a really fun trait to study. And you might be wondering, wait, do yeast age? They're a microbe. And they do. Microbes, like yeast, do in fact age. And yeast is an especially useful microbe for studying aging because it divides asexually anyway by budding, right? So what happens is that when a yeast cell divides, it produces a bud, which is called a daughter cell, and that leaves a scar called a bud scar, which is evident as one of these green glowing rings that's really chitin dense. And the more times that a mother cell divides, the more bud scars she'll get. And it's thought that on average, a yeast cell will survive 30 to 40 cell divisions before dying. And so there are a few ways that you can separate young from old yeast cells in the lab. Some folks use microfluidics devices, which are very low throughput. Some folks use engineered genetic switches. So like you can turn a gene on or off that will kill a young cell or an old cell, depending on the context. But we can't use those because we are focused on genetic diversity. And so the method that we've been using is bud scar staining. And this is work that's been done both by former postdoc Ian Cooch and current PhD student Katie McHugh. They spent a lot of time working with this bud scar staining technique where they can dye a population of cells with this fluorescent dye that binds to bud scars and then use a cell sorter to separate out the old ones from the young ones. And thankfully, as of a couple of years ago, we have a cell sorter here on campus. We didn't before that. Katie will tell you all about that. So it exists in this biomedical characterization equipment shared facility. And even though, so the PI on the grant that got it here is in chemical engineering, but I was involved in the team of folks who got it here. So I'll tell you a little bit about some of the work Katie's been doing. Basically, what she has done is she's worked a lot with our 4 founder population. She'll stain it, she'll put it through one of these sorts on the cell sorter, and she'll designate channels to isolate fractions of cells that we think represent the young and the old. We typically exclude the most extreme part of the distribution of cells because dead cells will autofluoresce and appear more fluorescent than the rest. But any cell in this sort of pink fraction would be old because it has a lot of bud scars and therefore more fluorescence. And any cell in this blue fraction might be quite young with one or just a couple of bud scars. And so we've shown, Katie has shown through lots of work on this, that this is a technique that indeed works. It produces cohorts of cells that are sort of their distributions are different in terms of the number of bud scars that we observe in these cohorts. So the differences are subtle, but they're definitely there. We get older cells in the old fraction, younger cells in the young fraction, and this was an initial concern because we wanted to make sure that bud scars was the thing we were really sorting based upon. And then what Katie did was she spent lots of time asking, do these two cohorts of cells have different properties with respect to their fertility, their sexual fertility? So she induced sporulation in young versus old cohorts and found that in fact, the young cells are more fertile, more able to produce spores than the old cells. And this is something that you might predict because of course, in many species like humans, sexual fertility tends to decline with age. And yet this is sort of an area of research that is growing in the yeast community. It's not really clear yet how universal this is, but we found that at least for our recombinant populations, it appears to be true. And this is really exciting for us because it suggests that late life sexual fertility is a trait that varies in our populations. And if it varies, we can select on it, right? And so we have proposed late life fertility as a selection regime that we could use in our yeast experiments. So in other words, we'd go through all the steps typical in our experiments, but in the selection phase, we would do cell sorting and pick the old cells, and only the old cells would get to reproduce. And so I don't really have time to show you the data that we've accumulated about this, but suffice to say the preliminary data is sufficient to motivate perspectives review article that Katie and I published last year, as well as garner interest from biomedical funding agencies. So both the Collins Medical Trust, which is a foundation that funds research at Oregon universities, and the NIH has funded this work. So I have a few minutes left where I can talk a little bit about where we're headed. I mean, of course, the aging stuff is ongoing. I expect that it will be a major theme of the research in the lab for the next several years, hopefully longer, but we're doing lots of other stuff too, that I'll just briefly touch upon. So one of the traits that we're interested in is in resistance to Roundup. This is a project led by former master's student, Chloe Hansen. Chloe is really interesting because she was an organic farmer for many years prior to coming to grad school. She owned her own business in Oregon. And so she knew a lot about Roundup and its potential impacts on the microbes in soil and soil health and how this is a growing concern for farmers. And so that was a trait we identified as one that would be cool to sort of team up on. And so even though Roundup is not supposed to be harmful to anything but plants, we think there's evidence that it is. And it turns out that, so she did an experiment where she subjected yeast populations to different dosages of Roundup. And the take-home message is that she found that as this PCA plot shows, we see distinct clusters related to dosage. So the genetic base, there is a genetic basis of Roundup resistance in yeast, and it's different depending on the dosage of Roundup used. And so Chloe has since graduated and moved on to a PhD program at UCLA, but we're continuing to work together to publish this work. And then another project that has just started in the lab is led by current postdoc Megan Sandoval Powers. And we are leveraging her expertise as a biochemist to think about how complex antifungal drug resistance might evolve. So Megan has chosen 3 antifungal drugs with different mechanisms of action, and she's studying how yeast populations evolve to them separately and together. And while this was admittedly at least partially motivated by all the craze last year about The Last of Us, where the fungi won, right? The HBO show and video game. It also is motivated by the fact that, clinically, practitioners often rely on cocktails of antimicrobial drugs to treat infections. And we are sort of interested in learning more about how microbes might evolve resistance to those cocktails and if those mechanisms might be different than how they evolve resistance to single drugs. And then finally, I'll just highlight some ongoing external collaborations I have. Again, I think the model system that we've developed has, it's attracted some interest from like-minded folks in the community. So for example, my longtime colleague, Elizabeth King at the University of Missouri, and I are working on combining theoretical and empirical approaches to model eco-evolutionary dynamics to predict how populations will evolve to climate change. So as I alluded to before, I think sometimes population genetic models, the traditional ones that we teach in the textbooks, are oversimplified, right? Of course they are, they're models. But one thing that they tend to assume is that selection is constant. And we know that's not true in the real world. For example, there are seasons, right? And populations in the real world have to deal with seasons. And with climate change, of course, it's not just one stress getting more stressful, it's the seasonality of stresses where environments are becoming more and more extreme. And so Dr. King and I are working on sort of combining in silico modeling approaches and mathematical theory that can be tested by our yeast experiments where we say fluctuate selective pressures and see how well they match. Another collaboration that we've embarked upon is with Dr. Katie Heil at North Carolina State University. She's an expert in yeast recombination rate evolution. So yeast is a fantastic model organism for studying meiotic recombination because they do it a lot, way more than humans. And so she was interested in our populations that have been selected for high versus medium stress, different selection intensities, because she is curious about how those selection pressures might have remodeled the recombination rate machinery or the processes that govern recombination rate variation in yeast. And then finally, Mike mentioned that, I started my work with Drosophila and I've continued to work with and collaborate with Drosophila folks. I just didn't have any time to talk about any of that stuff. But I have a long-term collaboration with my colleague Parveen Shahrastani at Cal State University Fullerton. And for the last 6 years or so, she and I have been working to evolve populations of fruit flies that have both postponed fertility and therefore longevity, but also enhanced innate immunity. So we have created, mostly her, it happens in her lab, she has created a set of selected lines that not only live longer than the ancestor or the controls, but they survive infection with entomopathogenic fungi. So they are more immune competent than your standard fly. And so we've been really interested in dissecting the genomics underlying both of these traits, longevity, as well as late life fertility, but also innate immune competence. And that's work that current PhD student Giovanni Cristani is working on. And Hannah Dugo is a former undergrad in the lab who has recently transitioned to the master's program here, and she has been working on that too. So, oh my gosh, I must have been talking really fast because I'm somehow on time. Sorry, but I'm done. So I just want to acknowledge the many, many, many people who have helped me do this work over the last several years. Like I said, it's been a huge privilege to get to do this work with such wonderful people and collaborators. And thanks to my two favorite people right there who make life outside of the lab fun as well. So with that, I will take your questions.
With your cryo studies, I'm curious how, what the freezing process was, if it's like flash or slow, because I can imagine you could get pretty different selection pressures on that process, like the osmoregulation process, depending on how you do it.
Absolutely. Yeah, that's a great question. So the way we did it was sort of selfishly motivated in that we basically just wanted, our primary motivation was, is this affecting us? Right? Is this affecting our sort of precious yeast populations that we work so hard to build that have all this diversity? So we were mimicking the conditions we'd use in the lab to freeze the stalks. And so that would be slow freezing. So we just put them in the freezer. We just sort of resuspend them in glycerol and then put them in the freezer where they freeze slowly. But I could imagine that, yeah, if you chose to flash freeze them and liquid nitrogen or make a slightly different choice, you probably would get a different response.
Yeah, like metal shuffling is a really fast response, but it's probably not ideal for cells to freeze. So that's interesting that you got that magnesium transporter or whatever it is.
Yeah, well, we'll see. I'm hoping we follow up on that work. We just haven't because Islana, graduated with her master's. But what we really need to do is prove that that's the gene that really matters because we haven't proven that yet. We just have suggested that.
Yeah.
I think I saw Aisha in the back first.
Yeah, when you are talking about stress in the cryo and the ethanol, what are you referring to? Like, what does stress mean for yeast?
Yeah, that's a good question, actually. So yeah, I tend to use stress and tolerance and resistance sort of interchangeably, though I shouldn't. When I say stress, I mean that not all yeast cells can survive it, or at least not all the cells in our diverse populations. But the mechanism of survival is often unknown. Like, are they tolerating the stress or are they resisting the stress? We usually don't know the answer to that.
So when you said they're more stressed, you meant more die.
More cells die. Yep, exactly. Ricardo.
So we have the question, what are the differences of using a population that has had 4, 8, or 12 founders?
Oh, sure. Well, ultimately, it's just the reservoir of genetic variation that we're starting with is different. And so in a perfect world, what we should do, if we want to say something truly general about a trait or an evolutionary process, is use multiple of those founder populations and see how similar responses might be among them. But in practice, we tend to just focus on one because we're human beings and these experiments are hard and we have to do one at a time. But yeah, really it's, they're there because I could imagine lots of scenarios where we might want to use one over the other, like say the 12 founder population has a lot more rare alleles than the 4 founder population. And so if you were interested in a question about selection on rare alleles, you might choose that one. Francis.
Well, thank you, Scott. That was awesome to see this. I'm really intrigued by this idea that you pose that populations get high centered, that they do well in the moderate, and everybody does well, but then you increase the stress, they do worse. Do we see that in populations a lot in the wild?
I don't go outside, Francis.
Oh.
No,
You're finding that sweets are rare. Maybe they're not high centered. You never lose the alleles that do really well.
Yeah, so the question is, well, I guess I'm not sure if this is what you're asking, but one thing we looked at was, do we get more selective sweeps in the high stress treatment versus the medium stress treatment? And the answer is no, we don't get sweeps. We really see little evidence of sweeps in either treatment. And so I don't think that the sort of selective sweep depends necessarily on the strength of selection. As far as why we get alleles that have different impacts in high versus medium stress, that's something we're just sort of starting to explore now as a result of this work. I think it probably has a lot to do with just trade-offs, right? Trade-offs that exist in populations. Maybe there's an allele that, I don't know, related to cell. I could imagine, I guess, a single allele that would have a consequence in medium stress that would help with the ethanol, but a different consequence in the high stress that would impede growth rate, right? Or in other words, there could be these pleiotropic effects where there are multiple stresses that are affecting the populations, and single alleles might work in one direction in one environment and a different direction in another environment. Mike.
I was quite fascinated by the large number of loci that seem to plateau, and then you find some kind of balancing selection. Have you or any of your students ever considered like experiments on any of those individual loci to see if you have like over dominance or anything going on?
Yeah, Yeah, I mean, well, to answer your question, we had one, that was the subject of one of my honor students' theses, but we didn't really see strong evidence. You know, we couldn't basically find the one allele that had a clear overdominant effect. So I think the tricky part is that we have so many candidate alleles that we're interested in to follow up on, either to validate their functionality or to maybe validate a heterozygote advantage, is that we don't know where to start. There's thousands. So that's the tricky part, is in figuring out which one to work with. But yeah, I would love to do that. And that has been done in certainly other work in yeast and flies, but usually it's in the context of a single mutation that had arisen and had a really large effect. And then you could validate its, heterozygote advantage. But we haven't been able to narrow things down quite so cleanly.
Great talks, very interesting. I think my question is kind of related to Francis, and you might have already touched on it, but I was wondering if you've seen or if you think you would see the same pattern as the ethanol experiment where you have like different responses in medium and high, if you would see that with other stressors where like maybe the medium would be response to a specific thing and then the selection on the high end would be more like overall survival. Do you know if like you would see maybe a similar pattern in, I don't know what yeast respond to, but like ethanol versus some other thing that you would expect?
Yeah, I mean, the answer is, yeah, I do think that now. And the one other result that makes me lean toward that as a general, likely truth is that the experiment I mentioned with my former student, Chloe. So the stress there was Roundup, which is a chemical that kills the yeast in some fraction. And her selective treatments were either one strong dose continuously versus a dose that started off really small and then increased gradually over time. And so it's the same chemical, but again, the intensity of selection was varied. And indeed, we see in the genetic evidence that that led to the recruitment of different variants, different genes. And so, it might be a statistical power issue and that we're only, maybe there's more commonality than it seems, and we're just not detecting things until selection is stronger. But at the same time, I think that a lot of times, if we want to really model the process of evolution in a way that's relevant to real populations experience more modest stresses. And that's what we should be focused on, not the super sort of the stresses that are almost comically high just so that we get a response. And so I think to answer your question, yeah, that's my view at the moment.
So in your sort of about the system in general, do you induce the sexual recombination just once at the onset of the experiment and then let them sort of asexually reproduce after the selective pressure has been applied, or is it constantly like reshuffling around as the experiment is going?
Yeah, good question. So the most common experiment type that we have, yes, we sort of generated that genetic variation in the beginning, and then we continue to shuffle as the experiment goes on. That's that sort of rainbow plot where we go step 1, 2, 3, 4, because, I guess a caveat is it's impossible to make the yeast have sex every generation because yeast don't work that way, right? They have to grow up a little bit asexually in between. So what we actually achieve is this pretty complex life history that involves periods of both budding and these like weekly, we call it the sex on the weekends protocol, because that's what it is. Every week we do this induced outcrossing protocol. And so that is a compromise in the sense that this is not, these yeast are not evolving in the same way that an obligately sexual organism would. At the same time, like I said, they're so good at recombination, they recombine more every meiosis than say a human would. And so there's actually, functionally speaking, about the same amount of genetic shuffling that happens as a result.
What do you mean when you say selection is not constant?
Well, I guess in the context of that the Roundup experiment, the dose of Roundup was increased, which means that the stress on the population was increased in a regular fashion. And so when I say that the traditional population genetic models assume that selection is constant, they're assuming that you can estimate a number that's like a selection coefficient that a certain proportion of cells die as a result of this stress every generation. But in the real world, I don't think that's how things work. And so with the Roundup experiment in particular, we were trying to model exactly that. Like what happens when you intentionally vary the strength of selection. Does that answer
Actually,
your
you
question?
mimicking the natural system, because in natural system, the selection is condition dependent, right? And based on genetic variability, the selection is different, right? It's not constant. I mean, I just want to be clear. So in the natural system, in the real world, selection is not constant.
Right. That's what I think.
Yeah.
Well, I mean, because of things like seasons or just variability, stochastic change. And so it's rare. So I guess we do all these evolution experiments thinking we're going to learn something about how populations evolve in nature. And yet when in nature is a population of, say, fruit flies going to experience the same exact selection pressure every generation for 500 generations? It just doesn't work that way, because they're in the lab. So I guess that's the distinction I'm trying to make, is that we're working toward building more complicated evolution experiments with more dynamic selection pressures involved, so that with the hope of one day making more concrete connections to the real world. We done? I think we're done. Thank you. Around if there are any lingering questions.